States. She's from the federal government of United States. Her presentation is on eco-friendly, innovative approaches to coffee leaf pest management in Hawaii, USA.
Thank you very much. Aloha, everyone, from the lovely state of Hawaii in the United States. I thank you so much to the organizers of this conference for inviting me to speak and share the research we've been doing. And hopefully you can see my slides and hear me okay. Before I get started, I would just like to thank my incredible team that's in the laboratory. You can see all their faces. They've done incredible work throughout these years. I really want to acknowledge all of the growers and the producers that we work with, which we couldn't do our research without them. I'd like to thank our colleagues at Ag Research Service and our collaborations at HARC, the university, and with SHAC. So to get started, in case you're not familiar with coffee leaf rust is caused by the obligate parasitic fungus Himalaya vistatrix, and its only known host is coffee, and it needs a living host to survive. So for infection to occur, pretty much there's the optimum water conditions, temperature, minimum, maximum, and the spores will actually land on the surface of a coffee leaf. They will work their way to the underside so they can enter through the stomates. They produce haustoria or specialized feeding structures, and then they actually germinate back out through the stomates and start producing chlorotic spots and spores. So once infection begins and the spores that you're looking at here get into the leaf, within about 10 to 14 days, you can start to see new spore production. So it's pretty incredible to think that a spot, the infection process takes only a few weeks, and within 3 to 5 months, approximately 300,000 spores are produced. And this pathogen survives mainly as mycelium in the living tissue. Okay, so Hawaii is the most remote archipelago in the world, about 2,500 miles from any landmass. But Hawaii was actually the last coffee growing region of the world to get coffee leaf rust. It showed up on Maui in late October 2020, and within about 8 months, it had spread throughout the state to the 6 main islands producing coffee. We were involved as a plant pathologist with the early detection and diagnostics, working to figure out the genetics and the race, because there are more than 50 races available throughout the world. And we started working on how to manage this problem through various fungicide testing, variety testing, and natural enemies, which are the last two I will give updates today. So to characterize the pathogen in Hawaii, as I mentioned, we had to set up not only prove pathogenicity, but we developed methods for rapid early detection, including a new qPCR method for Himalaya vistatrix. We were very fortunate to work with experts throughout the world, genotyping with Dr. Aim at the University of Purdue to figure out where it might have come from, where its closest related cousins, what part of the world. And this was shown to be probably from the Central America or Caribbean region, like Jamaica or Honduras. And the genetics showed it was actually a single introduction, so first into Maui and subsequently spread throughout the state. We also worked with the experts at the University of Lisbon in Portugal, Dr. Barzea and his team, to identify the race, which was determined to be race 24, containing virulence genes 2, 4, and 5, which are very pathogenic on varieties of coffee such as Typica, Bourbon, Catuai, and Caturra. And considering that Typica is the most popular and widely grown variety throughout the state, everything was highly susceptible. Okay, so one key component, of course, fungicides are important. We had very few registered for use on coffee, so we knew this would be a long-term project to look at varieties, but a critical component in the overall IPM strategy. And we started working not only in the lab, but using potted plants and doing studies, better understanding how the pathosystem was working in our environment in the field. So first and foremost, we developed and optimized a leaf disc assay for rapid screening, as well, considering we don't have outdoor space, we're trying to, as part of a germplasm repository, keep coffee germplasm from around the world clean. So the space we had available would be incubators or growth chambers. So we wanted to optimize how many we could handle at a time. And so for this assay, there was really four main stages. The first being the inoculum preparation. Because you can't culture this fungus, you have to actually go out into the field and collect new inoculum every time you want to work with it. And you could collect leaves. We'd go to the field within a day or so before we wanted to start the experiments, scrape the spores off, and create an inoculum concentration of approximately 1 times 10 to the fifth per mil. We would also then collect leaves. And we also, as part of our big collaboration, would have leaves sent to us from Oahu to Hawaii Island. And we'd go through the process of surface sterilizing them, preparing them actually as leaf discs using a cork borer. And this picture here shows the setup of this assay. And then you would apply the inoculum essentially as a liquid drop onto the surface, seal up the plates with high humidity in these plates in the dark for 48 hours, transfer to the light, and start taking data weekly for 4 weeks post-inoculation. Now, we were doing severity ratings based on a rating scale of Tomeo, which was essentially 0, no reaction whatsoever, to highly severe necrotic, chlorotic, with abundant spore production. Okay, so as I mentioned, our partners on Oahu with HARC, a Dr. Shifumi Nagai actually started a CLR breeding program at the late 80s, early 90s. And this was taking all of the farmer favorite accessions for each island, like Kona Typica here on Hawaii Island or Katuai on Maui, and crossing them with CLR resistant catamores. And this material was developed, actually planted in a field on Oahu, but screening could never take place locally because the pathogen was not here and not something we wanted to bring in. So they started sending material to us, and we started screening this material as rapidly as possible. And this is just this graph to show you. I know you cannot read it, but the x-axis, this is a subsample, the various accessions, and on the y-axis, it's how much sporulation. So if it's the pathogen is sporulating, the severity essentially is increasing, and we definitely get a good variety. Yes, we have some of the controls to the far right, super susceptible, producing a lot of spores, but as you can see, there are many that would maybe have some spots, but not actually produce spores. So this was kind of a highlight or bright spot to continue in the future. And this is The example of the leaf discs on top from the highly susceptible showing chlorotic spots and numerous spore production to an intermediate resistance in the middle with essentially just chlorotic spots but no spore production, meaning the pathogen was not completing its life cycle, which still would be very important material to look at, to many examples of highly resistant where there was no symptomology that would develop at all. And looking and as of this month, testing over 650 crosses, we see at least 500 showing resistance to coffee leaf rust, which is very, very promising. Okay, so ramping up, we wanted to utilize the space as much as possible and also prioritize what we were looking at for additional testing, moving particularly with the goal into the field. Okay. In the meantime, there are almost 800 small farms on Hawaii Island alone, and the incidence and severity, while all areas were affected by coffee leaf rust. Here's an example of this tree here, completely defoliated, and using the leaf disc assays just to show what's happening in the field. Now we can look at in the laboratory. This is the very susceptible typica, which we also see symptomology on Bourbon variety. This is what is widely planted all over. But we would find in a few farmers' fields these gems where you have this defoliation, but suddenly there was a few trees that would stand out that looked great. And so we started testing this material in the laboratory to see if it was also proved to be either highly tolerant or resistant. And we also partnered with the university, who had a large variety of catamore crosses, another susceptible to, with typica, the widely planted material. And so to show you the leaf disc assays on top, the very susceptible control, and these catamore crosses showing little to no symptomology. And this was seen, so not only in the field over 2 seasons, but backed up and leaf disc assays in vitro. So to summarize the first part of this research for variety screening, we saw a wide gamut from highly susceptible to highly resistant, which is very promising. And the results generally correlate to the field observation. So if it was highly susceptible in the field, we would also see high susceptibility in the assay. And resistance would show little to no level of infection, not only in the field, but proving this with leaf disc assays. Okay, and then I'm going to change gears to one of my favorite parts of the project, looking at biological control, actually looking for natural enemies to control coffee leaf rust. And this picture here shows you the various districts separated by color of Hawaii Island. And we conducted a microparasite survey at 22 different farms and field sites throughout the island early on in the process of when CLR started really with widespread distribution on island. And we looked at a variety of locations in the Kona, Kau, Hamakua, and Hilo districts, which is kind of north, south, east, west of the island, varying in elevation from 200 to 700 meters, a variety of rainfall, virtually none on the west Kona side, to 236 millimeters on our side of the island, Hilo side. Also, the farms being partial shade to full sun. We would collect leaves that had this suspect fungi associated with the CLR spores. We would scrape it, plate it onto half-strength acidified PDA or a CTC4T medium, which is selective, and go through the process of purifying, identifying using morphology and molecular barcoding, and within essentially a 1.5 to 2 year period, we sampled over 600 lesions from 200 leaves, resulting in 194 isolates to move forward with, which were part of 50 taxa and 44 different genera. And this is just to show you a picture of just the incredible diversity that we were isolating. Um, so the potential of these mycoparasites for coffee leaf rust management. So we started with our leaf disc assays, which worked very well, taking infected leaves from the field, creating leaf discs that had a spore production, and then seeing the efficacy of various isolates on these discs. And so we would then inoculate individual isolates onto the spores, the whole spray the surface of the leaf, and then take pictures and observe for pustule colonization over time, and actually calculate severity, which is the percentage of pustule colonized by the parasite, with pictures using Assess 2.0 software. And so the negative control in the top left shows you this abundant spore production, and these other potential mycoparasites were very good at colonizing, particularly the areas where spores were located on these discs. Just to look at it a different way, on the x-axis, the time, days post-inoculation versus mean severity on the right, the y-axis, and different genera and isolates. And within essentially 3 to 4 days, we would see complete colonization and almost complete overtaking of the growth of Himalaya within the week as compared to a no spray control. Okay, so we also then wanted to look at it. So that was one thing in a way to see the potential of these isolates to attack the spores, but could we actually use these isolates to protect the leaves from becoming infected in the first place? So we would collect healthy coffee leaves. We would bring them back, prepare our leaf disc assay, inoculate the surface with a fine spray of an individual isolate of a potential mycoparasite, and then a day later inoculate with Himalaya vestatrix. We would then collect data every week for 4 weeks post-inoculation, once again looking for chlorosis, sporulation, using the same severity ratings. And this slide shows the top left, the positive control, the negative control on the top right with no inoculum, and then 4 different isolates showing great potential. BL82 on the bottom right had a lower incidence and severity and very little spore production. And these other 3 isolates actually there was no spore production. So maybe some chlorotic spots were produced on some of the discs, but no spore production. So there would not be any additional disease outbreaks if this were applied. Okay, so as far as the next steps for these mycoparasites in the lab, we want to show that they are inhibiting actual spore germination. We want to do more work with kind of a pre or a post-infection control, again, for protection versus a curative effect. Try to look at combinations of fungi, and we definitely want to see if any of these isolates potentially have any adverse effect as a plant pathogen, we would rule those out immediately. Our goal is to now move from leaf disc assays. We've already started potted plant assays in a greenhouse, in growth chambers. Ultimately, we hope to see what possibility it is working in the field. And could it serve as a valuable tool for coffee farmers as part of an IPM strategy? So I'm going to end with really just trying to control coffee leaf rust in Hawaii. We're managing it as a continuous epidemic. There is no silver bullet. We have to use an IPM strategy with short-term and long-term strategies, including just optimizing plant and soil health, early accurate detection, using the tools we have available for fungicides, coppers, some of the biologicals, a translaminar, and we're real hopeful that there's promising results of resistant varieties and potential biocontrol. And we're definitely trying to avoid the introduction of new races or the evolution of new races which could overcome resistance. And with that, I will say thank you very much. I really appreciate the opportunity, your time and attention. I know it's kind of late there throughout the world. I'm happy to answer any questions, or please reach out to my email anytime if you have questions or want something clarified. Thank you very much.